Okay, continuing back to eosinophils. What do we have? We have the defend against the helminth. Very important. Helminth infections. Major basic protein. Bilobed, two lobes nuclei, packed with a large eosinophilic granules of uniform size. Look at the picture A. Large pink granules of uniform size. These pink granules are of uniform size. Highly phagocytic for antigen antibody complexes. They produce histamines, major basic protein, helminthotoxin, okay, and eosinophil peroxidase, eosinophilic cation and protein, eosinophilic drive neurotoxin. Eosin pink diphilic loving. Pink loving, eosinophilic, causes eosinophilia. Very important. These factors are important. What causes eosinophilia? Eosinophilia increase in eosinophils. Parasites, asthma, chronic adrenal insufficiency, myeloproliferative disorders, allergic diseases, neoplastic, specifically in the Hodgkin lymphoma, eosinophilic granulocytosis with polyangiitis. Okay, so these are the things that cause that. And when we say parasites, we're talking about parasites. So parasites are too large to be phagocytosed. That's why we need the eosinophils. Too large to be endocytosed or phagocytosed. Okay, and yeah, that's pretty much all we have. How do you stain them? To stain them, you can use with white stain. So these granules will stain red with white stain. All right, moving on with the basophil blue cells. What are they? These are they mediate allergic reaction. Densely basophilic granules, looking at densely basophilic granules, contain heparin, which is anticoagulant, and histamine, which is a vasodilator, and leukotrienes, which are synthesized and released on demand. Basophilic stains readily with basic stains. Basophilia is uncommon. Basophilia, meaning increase in basophils, is uncommon, but can be a sign of myeloproliferative disorders, particularly CML. Mast cells. The immediate local tissue allergic reaction contains basophilic granules, basophilic blue granules, originate from the same precursor of the basophils, but are not at the same type. They can bind to the Fc portion of the IgG to membrane. Activated by tissue trauma, C3a, C5a, and surface IgE, cross-linking by antigen IgE receptor aggregation. Okay, which leads to the degranulation and release of histamine. So let's say you have this, you have some antigen, and the IgE binds to it. But there has to be two IgEs. My bad. You have a cell which has the IgE and the antigen binds to those IgEs. These has to dimerize. They have to aggregate, like attach with each other, in order for the activation of this mast cell. So this is the mast cell, and it has IgEs on top. IgEs. An antigen coming here has to bind both of them, and these have to aggregate in order for the activation that leads to activation of this mast cell. Then this degranulates and releases histamine, heparin, and all of whatever it contains. Histamine, heparin, tryptase, and eosinophilic chemotactic factors. Excuse me, guys. It is involved in it is involved in type 1 hypersensitivity reaction, right? Very important. Chromoline sodium is a drug that prevents the mast cell degranulation. So, mast cell was going to degranulate as soon as it's stimulated, right? So, chromoline sodium inhibits that. It is used in asthma prophylaxis. Okay, vancomycin, opioids, and all the radio contrast dye can elicit IgE independent mast cell degranulation. So although normally we know that IgE leads to the degranulation of mast cell, they're saying not only that, man, what else? They're saying vancomycins, opioids, radio contrast dye, they can also elicit mast cell degranulation independent of IgEs. Now let's talk about mastocytosis. Mastocytosis, it is a rare proliferation of the mast cells in the skin and or extracutaneous organs that are associated with the c-KIT mutations and increase in serum tryptase. Okay. And this mastocytosis, what we mean by that? We have increase in histamine. That a lot of increase in histamine causes flushing, pruritus, hypotension, abdominal pain, diarrhea, peptic ulcer disease. Now we're like, why this is occurring? It is because of c-KIT mutation and increase in serum tryptase. Again, mastocytosis, proliferation of the mast cells, wherever they are, associated with this mutation and increase in serum tryptase, which causes all of this. So that's for the mast cells. Now let's talk about lymphocytes. Lymphocytes refer to the B cells. Lymphocytes refer to the B cells, T cells, and NK cells. They're all lymphocytes. T cells mediate adaptive immunity, NK cells the part of innate immunity, round, densely staining nucleus with small amount of pale cytoplasm. So look, their cytoplasm is so small, very small cytoplasm, and their nucleus is almost like all the cell is nucleus. And these are lymphocytes. Then we have the NK cells coming from here. The NK cells. NK cells, they're important in innate. Immunity, especially against intracellular pathogens. NK cells are larger than the BT cells with distinctive cytoplasmic lytic granules containing perforin and granzymes that when released act on a target cell to induce apoptosis. Distinguish between healthy and infected cells by identifying cell surface proteins induced by stress, malignant transformation, or microbial infection. Again, what do? Very important. They distinguish between a healthy cell and an infected cell. How this guy's infected, let's say, and this guy's normal, this cell. How do you know this is normal and this is infected? By identifying cell surface protein, like cell surface markers. Okay. Induced by stress, and that change in cell surface protein could be induced by stress, malignant transformation, or microbial infection. And induce apoptosis, natural killer, is cells that do not express class 1 MHC, cell surface molecules. So what do? They induce apoptosis if the cells, they're not expressing MHC class 1. Again, induce apoptosis in cells that do not express class 1 MHC, cell surface molecules. Example, which cells would not have MHC class 1 on them? Viral infected cells, in which these molecules are downregulated. Excuse me. But you got the point here, right? And what are the surface markers to this guy, to this NK cells? We have the CD56, CD16, and that's all. And we have the little granules inside it and the nucleus. This is all NK cells. Now let's talk about the B cells. B cells mediate humoral immune response, originate from the stem cells in response to bone marrow, and matures in the marrow. Migrate to peripheral lymphoid tissue, follicles of the lymph nodes, white pulp of spleen, uncapsulated lymph node tissue. When antigen is encountered, these cells will be converted into the plasma cells, and those plasma cells are gonna may produce antibodies, as we've discussed that already. And not just only antibodies, also the memory cells. So next time that bugs comes in, the memory cells start producing the antibodies which were produced back at the time at the first infection. Can function also as an APC, B for bone marrow. Okay. Let's talk about T cells. What are the T cells? They mediate cellular response and originate from the stem cells in the bone marrow, but mature in the thymus. Very important. They mature in thymus. They need to get thymic education. They're good students. They learn some extra steps. So they differentiate into cytotoxic T cells, which express. They have the CD8 positive T cells, cytotoxic, and they recognize MHC1. And the second type is the helper T cells. They express CD4 and recognizes MHC2. Okay. The rule of 2 is a good mnemonic to remember that. MHC2 times 4, so 2 times 4 equals 8, right? And 1 times 8 equals 8. So what do we mean by that? CD4 will have an MHC2 to make an 8. CD8 will have an MHC1 for us to make an 8. That's just a mnemonic, okay? The point to remember is CD4 has MHC2, or let's look here. CD8 recognizes MHC1. 8 times 1 equals 8. And CD4 recognizes MHC2. 4 times 2 equals 8. Just a mnemonic that we need to get to 8. Whatever need to get to 8, just multiply, divide, you got it. Not divide, my bad, just multiply. Okay. Recognize and regulatory T cells. And third thing is the regulatory T cells. 3 types of T cells, 1, 2, and 3. CD28, which is a co-stimulator signal necessary for T cell activation. Very important. CD28 is the necessary for T cell activation. Most circulating lymph nodes are T cells. Most circulating lymph nodes are T cells. What do we mean by T cell? All of these. Alright, let's go to plasma cells now. We did talk about plasma cells, that the B cell from the plasma cells. So let's discuss some more here. Plasma cells are they produce large amount of antibodies specific to a particular antigen, they have the clock phase. I mean, what is the clock phase? Chromatin distribution. Clock phase, just known as fact, clock phase for the plasma cells. Chromatin distribution and the eccentric nucleus. Yeah, look at the nucleus, which is eccentric. Look, nucleus is on the side. Okay. Abundant RER and well-developed Golgi apparatus. They have a well-developed Golgi apparatus. Arrows in A. Okay, that diffuse thing that you can see over there, that's a Golgi apparatus. Amazing. It is found in bone marrow and normally do not circulate in the peripheral blood. Multiple myeloma is a plasma cell dyscrasia. Multiple myeloma is a plasma cell. That's why I remember they make antibodies. Okay, the plasma cell dyscrasias. That's all for anatomy.